Previously, in the amphibian embryo, we learned that maternal effects, such as veg-T and beta-catenin, activins and things like that, are specifying ectoderm and endoderm. And we learned that zygotic effects, like the nodals, are specifying mesoderm. We learned that endoderm induces ectoderm to become mesoderm. We discussed some of the signaling that's involved, and although I didn't call it out by name, we actually described the Newcoop Center and the beginnings of the Spemann-Mangold organizer, and we talked about induction versus the community effect. In today's episode, we're going to be talking more about the gray crescent. We'll talk about some other very interesting explant experiments, specifically those done by Spemann and Mangold, and we will describe, at least in broad strokes, what the organizer actually is. Okay, so we're going back to the gray crescent, and one of the things that I want to take a moment and point out to you, because I neglected to do so earlier, is that in the mid-20th century, Peter Newcoop, you've seen the word Newcoop and Newcoop Center on slides, but I haven't really called it out to talk about it. So Peter Newcoop was studying the organizer and the maternal effects in the vaginal region, and essentially figured out that what we now call Spemann's organizer, or the organizer, is actually induced by, we call it autoinduction at this point, but it's induced by maternal factors that are actually moved during cortical rotation. And the region that concentrates those maternal factors that will induce the organizer is called the Newcoop Center. Okay, so now you understand that term when it comes up. Now, we're going back to the gray crescent because I want to refresh your memories about an experiment that we talked about a couple of times this semester, and it's one that was done very early on in the 20th century, where we took, or where Spemann took, a cleavage embryo, two-cell embryo, and separated the blastomeres, all right? And I showed you this in a podcast, but I also pointed it out like the first or second day of class and said we would come back and talk about this in detail. So what Spemann did is, using a hair, he tied a loop around the embryo at the point where the first cleavage furrow was, and made a slip knot, and as you tighten that slip knot, you can essentially separate the cells. And what they noticed was that if you do this where both blastomeres have a portion of the gray crescent, you get two normal embryos. If, however, you draw the slip knot tight 90 degrees out of phase with the gray crescent, so that one blastomere gets the entire gray crescent and the other blastomere gets no gray crescent, and that's illustrated for you in the diagram here in the illustration, then what happens is you get one embryo from the blastomere that had the gray crescent that appears normal, and the other one is what they called a Baustock, or that's German for belly piece, and it's essentially a ventralized bit of tissue that has no dorsal structure at all. Okay, so one normal and one belly piece. And the normal was from the blastomere that had the entire gray crescent in it. So that's experiment one, and that's telling us that the gray crescent is required for normal development. And it's further telling us that something in the gray crescent is probably responsible for induction of the body plan in the rest of the embryo. And that's precisely what Spemann concluded. And he called this process induction, and he named this region the organizer because it seemed to organize the rest of the body plan. All right, so for the second experiment that I want to point out to you, again by Spemann, This is a really interesting set of experiments. We're going to look at two more. The first one here, we are taking, this is an early gastrula, an early amphibian gastrula, and this is an explant experiment. So we're taking a piece of tissue from one embryo and we are explanting it, putting it in an ectopic location in another embryo of the same state. So we have two early gastrulas. We take a piece of ectoderm that would have been a neural ectoderm, and we then transplant that to a more ventral region where we know that that ectoderm is non-neuroectoderm and is fated to become epidermis. And what we get is normal development. So the output there, that third embryo that's a neurula, has a normal neural plate. It's not missing any tissue. It's not got any extra tissue in the neural region at all. And it's got normal epidermis, or normal non-neuroectoderm, in the non-neural plate region. So what's happened here is, even though we took some presumptive neuroectoderm that should become neural plate and placed it into this host embryo, the rest of the developmental process took place such that that transplanted tissue was induced to become what it should be, which is epidermis. All right, so let's think about this. We take a piece of ectoderm that's supposed to be neural tissue. We put it in a region that is fated to become non-neuroectoderm, or epidermis. And what happens is the rest of the embryo, that region around the transplant, has in some fashion communicated to the transplanted tissue what its appropriate fate should be, and has in a sense rescued that transplanted tissue, and we get the normal epidermal ectoderm there. So the question that's at the top of the slide there, conditional or autonomous, is referring to how we would describe this. And this is an example of conditional fate. So the presumptive neuroectoderm doesn't have to be presumptive neuroectoderm if you move it to a region that is presumptive epidermis. It's going to become the appropriate tissue, so it's conditionally fated. Now the second experiment, very similar setup to the first one. It's an explant, transplant from a donor to a host. We're transplanting prospective neuroectoderm, and we are putting it ectopically in a place where we have presumptive epidermis. The difference is that we're not working with early gastrulas, we're now working with late gastrulas. So these are embryos that are just finishing up gastrulation and getting ready to undergo neurulation. And what do we see? Well, we get a very different result. What we have here is an ectopic neural plate. So we have the normal neural plate, and then we have a secondary axis, a secondary neural plate. So we have somehow, by transplanting that tissue, it was not able to be rescued. We're not seeing conditional differentiation or conditional fate specification here. What we're seeing here is that that tissue is now autonomous. In other words, the rest of the embryo was not able to induce that transplanted tissue to adopt its spatially appropriate fate. There's something different about the late gastrula. All right, so that tissue was autonomous in the late gastrula. Now, whether or not it's because the tissue that was transplanted can't be induced to change, or whether it's because the field of epidermal ectoderm in the host embryo is no longer able to signal the transplanted tissue, those are both interesting questions, and we'll get to that later. What I want you to understand now is we've got two different scenarios here where we're illustrating both conditional and autonomous fate specification, and the only difference here is the timing, all right, is the stage of development of the embryo. The early embryo, we see conditional fate specification, and then we lose a window of competence to conditionally rescue the fate of transplanted tissue, and what we see is autonomous development. So these are some of the most interesting and telling experiments that I can think of that draw in all of the things that we've talked about so far. So we've got signaling going on, obviously, telling different regions of the embryo, different cells in the embryo at different locations, what they're supposed to do. And we've got competence to receive those signals. And there's obviously a difference based on timing for signaling versus competence. All right, so here's a table that goes through the various experiments and illustrates this major difference between working with early gastrulas, which are permissive and allow for dependent or conditional development, and late gastrulas, which are essentially autonomous at that point, where you have independent development. All right, so if the donor tissue was supposed to be epidermis and you put it into a host region that is supposed to be neural, you're going to get neuro in an early gastrula, and vice versa. If the host region was supposed to be neuro and you put epidermis in there, you're going to get neuro. All right. Now, the opposite happens in the late gastrula. You have prospective epidermis. You place it where you're supposed to get. If you have prospective neurotissue and you put it where you're supposed to get epidermis, you're still going to get neurotissue. If you have prospective epidermis and you put it where you're supposed to get neural tissue, you're going to get epidermis. All right. So just pay attention to this and remember that the competence and the signaling have to be in sync in order for you to get the outcomes that you expect to get. Okay, now in this last experiment, this is the most famous of the Spemann-Mangold experiments. This is the one that was done in 1924 that essentially Hilda Mangold based her dissertation on, and actually 10, 11 years later, Spemann won the Nobel Prize for this. So this is the one that we talk when you hear Spemann and Mangold, this is the experiment that you think of. So what they did here is they took just the dorsal blastopore lip, all right, and they transplanted that to a ventral region, basically 180 degrees away from where it was in the donor embryo. So the host embryo now has a second piece of dorsal blastopore lip. It's just in the ventralmost region instead of in the dorsalmost region. And you all know what happens. In this case, you get two complete axes of development. You get primary invagination and formation of the gut where you're supposed to get it, and you get a secondary invagination with a secondary notochord, a secondary neural plate, secondary somite development, etc., etc. You have two organizers, and you end up with Siamese twins. You end up with newts that are conjoined at the belly, and you basically have two complete body plans there. And this is what Spemann and Mangold called the organizer. So this region of tissue, this dorsal blastopore lip, that is now called the Spemann-Mangold organizer, or the SMO, is a region of tissue that is signaling in some capacity, informing, instructing, organizing the development of the rest of the embryo. So we broke symmetry with sperm entry. We had our DV axis starting to be established by maternal effects coupled with sperm entry. And you know the story of the signaling factors leading up to the Newcoop center. I'm telling you that the Newcoop center induces the mesodermal tissue that's going to form the organizer. And then the organizer seems to have the capacity to organize the body axes of the rest of the organism. Now, another thing that's really interesting to note about this that I want to point out to you is that the same thing happens in chickens, okay, in birds. So you have, if you look at the blastoderm in an early chick embryo, a late blastula, and you transplant that region near Kohler's sickle, and you move that into a host 90 degrees out of phase, what you get are two primitive streaks. You get an additional induced primitive streak, and that's because that region where Kohler's sickle forms, where the node forms, where Henson's node forms, is analogous to, actually I would say it's homologous to, the Spemann-Mangold organizer. It's the same thing. Okay, so this is not just something that you see in amphibians. It is a process that is recapitulated across animals. Okay, now I'm going to show you a little movie to illustrate this. So there was Spemann and Mangold, and here's Eddie de Robertis, a younger Eddie de Robertis, doing that actual experiment. So we're under the dissecting microscope. Those are two frog embryos. And you can see using a very, very, very finely sharpened number 5 forceps and a tungsten microneedle, he's dissected out the dorsal blastopore lip, and now he is putting it in an ectopic location on the second embryo, patting it down. This looks a lot easier than it actually is. All right, and then, ta-da, we get the conjoined twins. All right. So what have we learned? Well, we learned that the Spemann-Mangold organizer, or the organizer, can recruit component cells or competent cells in surrounding tissues to form dorsal mesodermal structures. All right, I didn't go into that in a lot of detail because we will get to that, but that's essentially what's happening here. We also learned that there's some differences in competence and in inductive capacity between early and late gastrulas. And we learned that the dorsal blastopore lip is actually the region where those signals that are organizing the body plans in the amphibian reside. And last but not least, we saw that this is not restricted to amphibians, but it is a process that we see across the vertebrates. And we see the same thing in avians for that matter. All right, that's all we have for today. We'll see you next time where we continue talking about the organizer, but take a more molecular approach.